Hi everyone. My name is Fraser Stewart. I'm a PhD student from the University of Dundee in Scotland, and today I'll be speaking to you about ultrasound-mediated targeted drug delivery in capsule format for treatment in the GI tract. So I'll give a brief outline of what I'll speak about today. I will begin by speaking a bit about the Sonopill program, which my work is a part of. I will then go on to describe the proof of concept capsule design and some components. I will give a brief outline to the transducer characterization and the comparison of the different piezoceramic materials that was considered for the transducer. And I will then discuss some of the functional testing that was carried out with the assembled capsule. So as I mentioned, my work is part of the Sonopill program, and Sonopill aims to combine micro-ultrasound imaging with video capsule endoscopy for diagnosis and therapy in the GI tract. So you can see this image here. This is a mock-up of what the Sonopill may see. So the central image is what you would see from the video capsule endoscopy, and the micro-ultrasound scan is around the edge. So currently, diagnosis in the GI tract, one of the most common ways of diagnosis is using an endoscope. So an endoscope can be quite uncomfortable for the patient, and it can involve quite a skilled clinician to use the endoscope. So it's not ideal. So you can see an image of Sonopill to scale next to the endoscope, so it's far smaller. So that's not a speck of dust on the screen, that's the Sonopill. So there'll be some complex engineering involved in this. But anyway, my role in the Sonopill program is on the therapeutic side. More specifically, I'm working on ultrasound-mediated targeted drug delivery. So targeted drug delivery has advantages over other methods of drug delivery because a higher dose of drug can be delivered locally at the target, and adverse side effects can be avoided since it's delivered to the target. So the clinical research fellow in the Sonopill program has highlighted Crohn's disease as a potential disease for treatment. So Crohn's disease is an inflammatory condition in the GI tract. Mostly occurs in the small bowel, and it's currently non-curative, but medications for Crohn's disease have adverse side effects. So if we can deliver the drug locally to the treatment site, then it could reduce these side effects. So the work I'm about to discuss describes a proof of concept capsule for ultrasound-mediated targeted drug delivery in the GI tract. So this is a schematic of what the final therapeutic Sonopill capsule may look like. So you can see it's quite complex. It's got positioning systems. It's got quite a lot of electronics, cameras, antenna. However, in order to get some early results and some early testing, we can make a proof of concept capsule. So focusing on the components that would be most useful for targeted drug delivery, so obviously the drug delivery, the focused ultrasound, one of the cameras, and having the electronics controlled from out with the capsule, so it significantly reduces the complexity of it, adding in a tether, so reducing the ones that aren't important at the moment. So here's what the proof of concept capsule could look like, and it's tethered, so all these components are controlled externally from the capsule. So a bit about the capsule design. So the capsule was designed in SolidWorks, and it's been 3D rapid prototyped. You can see the 3D printed shell of the capsule on the lower left. The size of the capsule is 10 millimeters in diameter by 30 millimeters in length, which is comparable with capsules used for video capsule endoscopy. So the capsule consists of focused ultrasound transducer, drug delivery channel, CMOS imaging camera, and illumination. And I'll discuss each of these components in a bit more detail shortly. So each of these components are confocal at a point around 4 millimeters from the edge of the capsule. So this is the assumed location of the bowel wall. So a bit more about some of the components. So we'll begin with the camera. So you can see the camera is this green image here. It's 1.2 millimeters in diameter by 5 millimeters in length, and the company that makes it claims it's the smallest in the world, smallest CMOS imaging camera in the world, I should say. It's got a resolution of 220 by 224 pixels, so reasonably good for the size, and it's controlled out with the capsule via its own digital signal processor. So the camera needs its own illumination, so this consists of a printed circuit board with 4 LEDs in a ring pattern. You can see it on the lower left here, and this is controlled out with the capsule with a DC power supply. So now a bit about the drug delivery channel. The drug delivery channel consists of a fine bore polythene tubing with an outer diameter of around 1 millimeter. It can be seen on the top left image here. It's got red food dye in it just for visibility. And it's controlled out with the capsule also via a syringe pump. And the tether consists of a vascular catheter. It's got an outer diameter of around 2 millimeters. And each of the cables of each of the components is passed through this. And finally, a bit about the transducer, possibly the most important part. So we compared different piezoceramic materials for inclusion in the capsule. So we chose PZ26, which is one of the most common piezoceramics used in focused ultrasound applications, and PZ54, which is a relatively new piezoceramic used in focused ultrasound applications. So the properties of these piezoceramics, as stated by the manufacturer, is shown in this table at the bottom. They're quite similar except for the frequency. The frequency of the PZ26 is 3.5 megahertz. PZ54 is 4 megahertz. And the unfocused poles can be seen in this image here. So we fabricated the transducers with a microballoon loaded epoxy backing layer connected to coaxial cable. So the outer dimension of this transducer, fully fabricated, is 6 millimeters in diameter, 4 millimeters in length, and it's the coaxial cable is connected to BNC connector, and this is controlled via a signal generator. So to get more details about the transducers, we've carried out some characterization. So we characterized both PZ26 and PZ54. So we carried out some electrical impedance spectroscopy measurements and found the central driving frequency of both transducers was near that stated by the manufacturer, which was a bonus. And there was no impedance matching required either. So we then carried out some ultrasound field mapping. This was done in an acoustic scanning tank with needle hydrophone moved about in the ultrasound plane. So you can see the pressure map here. So driving the transducer at 4 volts peak to peak, we found an acoustic pressure of 125 kilopascals and a focal beam diameter of 0.75 millimeters. That was for the PZ54 transducer. And for the PZ26 transducer, we found an acoustic pressure of 157 kilopascals and a focal beam diameter of 1.35 millimeters. So we can compare both these transducers at this stage before carrying on out any more characterization. So the literature states that pressures in the range of 50 to 300 kilopascals is suitable for reversible sonoporation in the GI tract. So sonoporation is a process which is crucial in some targeted drug delivery applications. It's the process of using ultrasound to increase cell membrane permeability. So both our transducers have acoustic pressures in this range, which is good. And also both transducers have a small focal beam diameter. However, we've chosen PZ54 to be housed in the capsule because it's got a smaller focal beam diameter, allowing a smaller area of sonication and better targeting. So we've now carried out further characterization of the PZ54 transducer. So we carried out acoustic power measurements using a radiation force balance, and at 4 volts peak to peak, we found an acoustic power of 20.45 milliwatts. And we then carried out some thermal testing to make sure the transducer doesn't overheat during use significantly. So this thermal test was done using a thermal camera focused on both the transducer and on the coaxial cable. And it was done in air because if it didn't overheat in air, the idea is that it wouldn't overheat in water. So this graph shows the temperature of the transducer at different input voltages. And neither the transducer or the cable overheats significantly at 4 volts peak to peak. However, the temperature of the transducer began to overheat after this. So we carried out some functional testing of the capsule. So this was done using microbubbles passed through the drug delivery channel. So we monitored the behaviour of these micro bubbles at the ultrasound focus using the camera. So this video here shows the microbubbles approaching the ultrasound focus and abruptly changing direction due to the acoustic radiation forces. This could be a potentially useful result for targeted drug delivery applications because it could translate into drug-loaded microbubbles approaching the ultrasound focus and abruptly changing direction towards the target site. So a brief conclusion of the work carried out, we designed the therapeutic capsule for ultrasound-mediated targeted drug delivery in the gastrointestinal tract. And we highlighted Crohn's disease as a disease of interest that the capsule could be used to treat. The capsule consisted of transducer, and we compared different piezoceramics for capsule inclusion. So we compared PZ54 and PZ26, eventually choosing PZ54. The capsule also consists of camera, illumination, and drug delivery channel. We found two possibly clinically useful results with the capsule. That one was that the transducer has acoustic pressures which could be suitable for sonoporation. The other being the transducer able to push the micro bubbles towards the target location. So translating these two results into a targeted drug delivery application could be that drug-filled microbubbles approach ultrasound focus. The transducer then pushes them towards the bowel wall, and this tissue on the bowel wall is being sonoperated, increasing the cell membrane permeability, allowing these drugs to pass in deeper. So that's it from me. There's some acknowledgements. Basically, the Sonopill team, EPSRC for funding the project. Thank you for your attention. I'd be happy to answer any questions. Yeah.